Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing Central University's Common Entrance Test MSc Entrance question paper for the subject Applied Microbiology conducted in the year 11th March 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of Applied Microbiology, you can go to my channel Omni Bio Entrance. So in the channel playlist, you can go for CUET where you will find previous year question papers. Then next one. Which of the following statement is true for psychrophiles? Their enzyme have greater amount of beta sheet secondary structure. Not beta sheet, they have alpha helices. Alpha helices, they provide flexibility, they provide more flexibility than the beta sheets. So A is not correct. Their cytoplasmic membranes have higher content of unsaturated fatty acid. That is correct. Their enzymes have greater polar amino acid content. Yes. Their transport protein function at low temperatures. Yes. B, C, D only. Answer is option 1. The capsid symmetry of pox viruses is complex. Answer is option 3. Which of the following statement is correct? The terminal electron acceptor in anaerobic respiration is carbon dioxide only. No, the terminal electron acceptor varies in anaerobic respiration. You have nitrate as terminal electron acceptor in denitrification process. Then you have sulfate in sulfate reduction and carbon dioxide in methanogenesis. So that statement is not correct. Fermentation involves an internal inorganic electron acceptor. It is not inorganic, it is internal organic electron acceptor. So pyruvate, acetyl-CoA, oxaloacetate, fumarate are examples. So 2 is also not correct. Aerobic respiration involves complete oxidation of sugars. That's correct. The energy yield in anaerobic respiration and fermentation is same. I think there was a printing mistake for that. It should have been energy yield in aerobic respiration and fermentation. If that was the case, if it is like anaerobic respiration and fermentation, then fourth option is also correct because they have mentioned anaerobic respiration and fermentation. Fermentation is also anaerobic respiration, so fourth one is also correct. If it was aerobic respiration, then it would not have been correct. If aerobic means 36 to 38 ATP molecules per glucose molecule and fermentation only 2 to 4 ATP molecules per glucose molecule. Then which of the following is a professional APC? APC is antigen presenting cell, and there are 3 professional antigen presenting cells, that is macrophage, dendritic cell and B cells. So out of these, macrophage is the answer. For the separation of DNA by electrophoresis, which of the following method is used? Agarose, horizontal. Answer is option 1. Then which pathway replaces TCA cycle when E. coli is grown on acetate as the only carbon source? It is glyoxylate pathway. Answer is option 3. Dissimilatory nitrate reduction take place under? It take place under strict anaerobic conditions. Answer is option 1. Wobble hypothesis refers to the less stringent base pairing specificity of the. So wobble hypothesis, the 5 prime, okay, before that, you have got the 5' end of tRNA with G in normal pairing, and 5' end of normal tRNA with G, and 3' region of mRNA with C, that is normal pairing. But this position is likely to be a wobble position. Because it will allow the nucleotide at that position to pair with multiple bases in the mRNA codon. That is, here if you see, it pairs with, in wobble pairing, if you see, G pairs with U. So this position is wobble. So the 3' end of mRNA pairing with 5' end of anticodon is called as the wobble pairing. So 5' end base of the anticodon, that is correct. 5' end of the anticodon is pairing with 3' end of the codon. 5' end base of codon, no. 3' end base of anticodon, no. Middle, no. So it is the 5' end base of anticodon that is pairing with the 3' base of the codon. That position is called as the wobble position. Then GUG codes for valine in both prokaryotes and eukaryotes, but when GUG is initiation codon, it codes for. So it codes for methionine. Answer is option 2. Then arrange the following steps involved in bacterial transformation in correct order. So first will be binding and entry of extracellular DNA into competent cells, C. Then comes degradation of one strand of transforming DNA, pairing followed by integration of transforming DNA, and then generation of a transformed cell. So the sequence is CDAB. Answer is option 2. Then protein and their function. Trypsin. Trypsin is an enzyme, A3. Collagen. Collagen is intercellular ground substance, B4. Insulin is a hormone, C1. GLUT4 enables glucose transport into cells, D2. So the answer here is 4, A3, B4, C1 and D2. The correct sequence of enzyme action is, okay, binding of substrate to active site of an enzyme, B. Then binding of substrate induces the enzyme to alter its shape, A. Then breaking of the chemical bond of substrate and formation of new enzyme product complex, C. And then finally D, products of the reaction are released by the enzyme. So the answer here is option 2, that is BACD. Then next one. Select the correct statements with reference to cofactors of enzymes. They are non-protein constituents of enzymes that bound to enzyme to make it catalytically active. That is correct. Prosthetic groups are tightly bound to apoenzymes. That is also correct. The protein part of an enzyme is called the apoenzyme. Yes. Coenzyme NADP contain vitamin niacin. Yes. So all 4 statements are correct. Answer is option 3. Then which of the following is not a function of the human microbiome? Synthesis of vitamin B12, yes. Providing immunity, yes. Protects against disease-causing microbes, yes. Secretion of insulin is not a function of the human microbiome. Then next one. A single nucleotide pair is inserted near the 5' end of a protein coding DNA sequence. A single nucleotide pair is inserted in the DNA sequence. The most likely effect of this production will be, it will be a protein with an almost completely altered sequence, because this insertion, it will result in what is called as frame shift mutation. So it will result in a protein with almost completely altered sequence. Whereas first option, if you see a protein with single altered amino acid, that would result from point mutation, not a frame shift mutation. Then a highly truncated protein. A highly truncated protein would result from nonsense mutation or deletion, but not frame shift. So the answer here is option 2. When a mutational event leads to the replacement of one codon for another codon for the same amino acid, the resulting mutation is called, it is called silent mutation. Answer is option 4. Then mutagen and the function. Clastogen, it causes fragmentation of chromosome. Oncogene induces tumor formation. Teratogen results in developmental abnormalities. Ethyl methane sulfonate is an alkylating agent. So A1, B2, C3, D4. Answer is option 1. Then which of the following statements is or are not true regarding significance of meiosis? So it is a mechanism by which conservation of specific chromosome number of each species is achieved across generations in sexually reproducing organisms. That is correct. They have asked not true. So this cannot be the answer. Increase genetic variability, yes. Decrease genetic variability, it is not true. Variations are not important for the process of evolution. They are important. So C and D are the only answers. Answer is option 2. Then the primary target of HIV is, that is CD4 plus T lymphocytes. Answer is option 3. Then next one. The sequence of steps for live recombinant vaccine production is putting the cells in the presence of an inhibitor, transfection, formation of recombinant plasmid, expression of recombinant genes. Okay, so first one will be formation of recombinant plasmid. C first one means answer is option 4. Because option 4 is having C first. Then transfection of newly created plasmid into previously infected host cells. B. Then comes putting the cells in the presence of an inhibitor. A. And expression of recombinant genes. D. So C, B, A, D. Answer is option 4. Which of the following is an inhibitor of complex 4 of electron transport chain? It is cyanide. Answer is option 4. Key enzyme of the ED pathway. ED pathway is enter-Duodorf pathway used by bacteria to break down glucose. And KDPG aldolase is the key enzyme. It is 2-keto 3-deoxy 6-phosphogluconate aldolase, and it catalyzes the conversion of KDPG to pyruvate and glyceraldehyde 3-phosphate. So the answer here is option 2. Then which of the following types of cheese utilize Lactococcus lactis in its early stages of production? So mozzarella doesn't use, cheddar uses, Roquefort and Parmesan uses. So B, C, D only. Answer is option 4. Then which of the following is not a primary pollutant contributing to global air pollution? Carbon monoxide, yes. Sulfur oxides, yes. Sulfuric acid, it is not a primary pollutant. So 3. The oxygen liberated during photosynthesis comes from water. Answer is option 1. Then list 1 with list 2. Renin. Renin is for coagulation of milk. A2. A2 is there only in option 4. Then protease is used in household detergent. DNA polymerase is in PCR technology, and invertase is in soft center candies. So A2, B3, C1 and D4 are the answer.